So NfL has been called the CRP of the neurological diseases, right? So, and for those of you who are not familiar with CRP, this is basically indicating an inflammatory state, but rather unspecific. It kind of comes up, when something's happening to the body. A bit delayed, a bit unspecific always, and there's now much more accurate markers such as interleukin-6 also to indicate inflammatory states. Obviously also more expensive in clinical practice, but for neurofilament light chain, it has been called CRP for exactly this. It indicates very specifically neuroaxonal damage. It's a structural protein within the axons of neurons. And whenever there's, for whatever reason, for whatever cause, there is neuronal injury in the brain, you will have higher neurofilament light chain levels in your blood. That might be because of multiple sclerosis, that might be because of traumatic brain injury, because of stroke and so on. And we performed an experiment on with the blood of ischemic stroke patients, because we felt that potentially neurofilament is something that is we can analyze very specifically using a single molecule assay, and that has we can detect it early after stroke, but it did not turn out to be the case. And neurofilament after stroke kind of takes a delayed course in elevating. So in our analysis, we found that it peaks at day 7. Most likely it peaks even later after stroke. So also neurofilament is not a good marker for indicating early neuronal injury. If a patient comes in, how much is there lost in terms of neurons, how much is there to save? Neurofilament will not help us in the acute phase, but neurofilament is very accurate in determining in our analysis how much neural injury happened until day 7, let's say. And what we currently believe is that it's more accurate than neuroimaging-based markers, because still, even if we would define the size of an infarct using MRI, there's still many millions of neurons fit into one voxel, and neurofilament basically in the blood should tell you exactly theoretically how many neurons basically die. So what we currently believe is that neurofilament could have a case as a use of as a surrogate marker for treatment effects. So if we treat a patient in the acute phase, such potentially using thrombectomy, or whether we would use a neuroprotectant, there are studies also for this. And we currently need to evaluate in clinical studies the functional outcome of these patients at 3 months. This is scaled from 0 to 6, and obviously is very inaccurate and takes a lot of patients always for clinical trials to see their treatment effect. What we believe is that there's a case for neurofilament to be quantified at day 7 and to be much more accurate in depicting whether a treatment in the acute phase is helpful or not. This obviously needs to be shown across several studies, and we currently just started doing that. But I think there's a good case and a good chance of that being successful.